We often forget that we are covered in bacteria. Fortunately for us, most of this bacteria cannot infect us. We refer to this kind of bacteria as commensals. As you know, there are some bacteria that can harm us and cause infection. These are pathogens. There are some bacteria that can exist as commensals, but under certain conditions can become pathogens. This is what my lab studies. How do bacteria cause disease? We focus on one type of bacteria known as Staphylococcus aureus. Staphylococcus aureus does exist as a commensal. And about 70% of the human population, it lives right inside your nostrils, not causing any harm. But if you've heard of Staphylococcus aureus before, you've probably heard it in reference to staph infection, which is an infection of the skin. Or maybe MRSA, methicillin-resistant Staphylococcus aureus, which is a highly antibiotic-resistant form of Staphylococcus aureus that is increasingly harder to treat. Antibiotic resistance is a huge problem in the medical community today. Staphylococcus aureus also has the ability to infect many different tissue types, including the heart, causing endocarditis, the blood, causing sepsis, the phenoreproductive tract, causing toxic shock syndrome, the bones, causing osteomyelitis, and it can even cause food poisoning. How is it possible that one bacteria is able to infect so many different tissue types? To answer this question, my research focuses on sRNAs, or small regulatory RNAs, which are simple molecules that the bacteria can quickly produce in certain different environments. These molecules usually function in regulating different cellular processes like toxin production, which is necessary for a pathogen to do to be able to infect a host. My lab has identified one small RNA known as TEG41. When we genetically remove TEG41 from the cell, Staphylococcus aureus loses its infectiousness. We believe that TEG41 is essential in making the switch from a commensal to a pathogen for Staphylococcus aureus. We are really excited about the potential of small RNAs as they represent a new target for future antibiotics. If we can remove small RNAs from the cell, we remove that bacteria's ability to regulate those infectious-causing processes, meaning that we can essentially revert a pathogen back to a commensal. My name is Julia Tennant. I'm a third-year PhD student in Dr. Rodencarrow's lab, and I'm really excited and passionate about my research. Thank you all so much for your attention, and I hope you have a great night.